Welcome to our first podcast on lymphoid tissue. We will discuss and define the lymphatic system, but we will also break it down into different categories. We are first going to look at diffuse lymphatic tissue, and we categorize that as MALT. MALT stands for mucosal associated lymphatic tissue, and so we're going to be able to identify the different locations in the body where we'll have these. But then we're also going to look at the encapsulated lymphatic organs. And we're going to start in this podcast by looking at lymph nodes. So with lymph nodes, we're going to define the structural components, the different compartments of a lymph node, and then we'll describe the cellular constituents that you find that comprise a lymph node. So lymphoid tissue really is to function with the maintenance of fluid in the internal environment. Remember from physiology, fluid leaks out of our capillaries and not all returns. And so this excess fluid would result in swelling or edema in the tissues if we didn't reabsorb that. And what does not go back into circulation is captured by lymphatic capillaries and returned to our circulation. But also along the way, we have a number of organs such as lymph nodes that are going to present this circulating material or a interstitial tissue wash, if you'd like to think of lymph in that way. The pathogens that were washed out of the tissue and in the lymphatic circulation, they're going to encounter a very high concentration of leukocytes, especially lymphocytes, in these nodules and our nodular tissue. And so that's going to function in a means of the immune system. So as we say, plasma is filtered out of the blood and into our interstitial spaces. And this is where we change the name. It's no longer plasma, but we refer to it as interstitial fluid or lymph once it is absorbed back into our lymphatic system. So our lymphatic capillaries will drain this excess fluid and return it to the venous system at the level of the vena cava. Now, some interesting things. Lymphatic vessels are really going to parallel veins. Where you have veins, in all likelihood, you're going to see lymphatic vessels growing in ever larger and larger diameter. Lymph nodes are going to be located along the pathway, and we'll see an example of these. You have them in the cervical region of your body, your inguinal region, axillary region, all along the length following your alimentary canal, because these are some areas where you might have pathogens trying to gain access to the body. Now, within these encapsulated lymph organs called lymph nodes, we will have collections of lymphocytes that are called lymph nodules, and we will distinguish, we'll compare and contrast those. But within lymph nodes, you will have a number of lymphatic nodules. Now, not only are lymph nodes lymphatic organs, but we're going to see other encapsulated organs are going to be tonsils, thymus, and spleen, and we will cover those in later podcasts. Here you see a diagram of our systemic and pulmonary circulations. You can see the arterial flow in red, the venous flow in blue, and here we have the lymphatic flow illustrated in green. We have the lymphatic capillaries draining into our larger lymphatic vessels. Here we have an example of what the artist is trying to show as a lymph node, and within that we'll have lymphatic nodules, and you can see how this is illustrated to be returning to the venous system of the blood. Now, the first type of lymphoid tissue that we're going to characterize is referred to as diffuse lymphoid tissue or lymphatic infiltrates. What we're going to see is in a lot of areas where we have loose connective tissue, we can have collections of lymphocytes. Now, these are going to be transient in nature. They might not always be in exactly the same place, but in many instances, you will usually find them associated with the mucosal lining of hollow organs, and we'll associate that term mucosa, much like in our circulatory system we have the tunica intima. Well, mucosa is going to represent the inside layer of our respiratory and our alimentary canal. Now, if we associate this mucosa-associated lymphatic tissue or MALT in our gastrointestinal tract, then we can also refer to it as GALT. This is a more specific term, gut-associated lymphatic tissue. We can have it in our urinary tract, our genital tract, and if it's in our respiratory tract, we can refer to it as bronchi or bronchiolar associated lymphatic tissue or BALT. MALT is more of a general term for the mucosa in any of these areas. So here we see a slide that we're becoming more and more familiar with. We've seen this a number of times as we look at the lumen of our respiratory tract. If we look really closely, we could see our respiratory epithelium, which is our pseudostratified ciliated epithelium. We have our hyaline cartilage, part of our cartilaginous rings of the bronchi, and then we have some muscle associated with that. Well, if we look in this region below our epithelium, we're going to see that we have this very dark granular staining material, and this is a collection of lymphocytes. And so this is going to be our BALT, our bronchiolar associated lymphatic tissue. And it's in these areas where you will see these collections of lymphocytes. And again, once again, these are transient. They're not always going to be in exactly the same place and look exactly the same shape. So that's our diffuse tissue. Now we're going to move into our actual organs, and we refer to them as encapsulated because these are going to be bounded by a collagenous capsule that's going to define the boundaries of these organs. And in this podcast, we're going to cover the lymph nodes. Now in this illustration, you can see, as we mentioned before, where we have higher concentrations of these lymph nodes. We can see in our cervical region, the axillary region. There we have, following our gastrointestinal tract up to our diaphragm and liver, we have a lot of lymph nodes. And down in our inguinal area of our body is where we have a lot of these lymph nodes. And you can see the lymphoid drainage all draining back through these lymph nodes back toward the heart. Now, lymph nodes are going to be somewhat characteristically shaped. They're going to look like little beans in the body. They may be a few millimeters in length, or they may be up to 2 centimeters. So they're very variable in size. And these lymph nodules are going to be the place where we have this really high density of leukocytes. So we're going to have phagocytic cells such as macrophages and our granulocytes. They're going to be able to filter this particulate matter out of the lymph fluid. And they're also going to present these to other cells of the immune system, such as the lymphocytes. So we're going to have antigen-presenting cells. And once again, our macrophages are our chief APC that we have in a body. Now, it's very interesting because 99% of the material, we're not talking about the fluid volume, but the material that's present in the lymph, 99% of that is phagocytosed. And again, the vast majority of that is going to be phagocytosed by macrophages. Now, this is illustration. We're not looking at a lymph node in totality here, but we're looking at a section through a lymph node, and what I want you to appreciate are all the dark staining cells that we see near the bottom. This particular tissue section was produced, and before it was fixed, these cells were stained with an India ink or some kind of carbon black material, and then incubated at 37 degrees before the tissue was actually fixed in chemicals. And what this did was it gave the phagocytic cells time to incorporate this dark staining material. So everywhere you see this accumulation of dark material, that's going to represent a macrophage. And you can see the huge density of macrophages here present inside this just small field of an overall much larger lymph node. So with a lymph node, we are going to bring fluid, and remember, this is not part of our circulatory system, this is the lymphatic system, but we're going to bring fluid to the node, and this is going to be in our afferent lymphatic vessels, A for arriving to the vessel. These lymphatic vessels are going to penetrate the capsule and empty this material into the cortex of our lymph node, and this cortex is largely going to be subdivided into segments by connective tissue trabeculae. They're not going to be completely isolated, but around these trabeculae you're going to have areas and sinusoids where this fluid can flow in and around and percolate through all of our leukocytes and lymphocytes and macrophages that we have inside the tissue of the lymph node. This is all going to drain toward the center of the lymph node, or the medulla, and this is going to empty into the indented part of our kidney bean-shaped lymph nodule, and that's going to be the hilum. And from here, we're going to have afferent lymphatic vessels leaving. Now we will also have arteries and veins supplying the node, and they are also going to enter the arteries, and veins will leave at the hilum as well. So here we have our illustration. We can see an afferent lymphatic vessel, and there's going to be a number of these entering into each lymph node, and you can see our unidirectional cusps of the valves here for these larger vessels. Here we can see extensions of our connective tissue capsule extending down and subdividing, especially the cortex part of our lymph node, into smaller compartments. And the light area that you see, these are going to be sinuses. We're going to have a sinus that's going to be directly underneath the capsule. We can call this subcapsular sinus. We have sinuses that are going to go down beside the trabeculae. These are often referred as paratrabecular sinuses. And as this fluid is percolating through, think of all of the cells in here almost like the sand of a swimming pool filter. And these cells are going to be able to capture this debris, phagocytose it, as the fluid all drains toward the medulla. And here again we have medullary sinuses, the spaces between. These medullary cords are not structural, they are comprised of accumulations of cells. And so through and around these cells, the fluid is going to flow until we get to our efferent lymphatic vessel that is going to leave the lymph node at the level of the hilum. So as we saw in the illustration, we have a connective tissue capsule. It's going to be dense, irregular connective tissue. And beneath that capsule, we have a space, the subcapsular space. And this is where the lymph is going to leave the afferent lymphatic vessel and begin to percolate through all of these areas and all of these cells that we have inside of the lymph node. Now, these aren't simply empty spaces. The lymph node would collapse if we just had fluid-filled spaces, for instance. But we're going to have cells that are referred to as reticular connective tissue cells. And some of these are going to be reticular epithelial cells. Their job is to maintain the shape and the integrity of these sinuses. They're not going to be involved in immune responses. They are simply there to hold that space open. And so in our illustration here we have our capsule of a lymph node. And you can see this space. We do have some cells in here, and most of these cells we're looking at are going to be our lymphocytes. Some of the lighter staining, larger ones will be macrophages, but we can't make out that detail in this particular image. But this space that's supported by these reticular cells, this is our subcapsular space, our subcapsular sinus. And in this instance, as this space dives down, we're likely going to see a trabeculae extending down here as well. This is going to extend downward as a paratrabecular sinus. Here's another view of the capsule of a lymph node. And what we've done is we've cut a cross section here, almost longitudinal section through a lymphatic vessel. Since we're in the capsule and this is running in and along, this is going to be one of our afferent lymphatic vessels. And this material is going to get dumped down into the cortex of this particular lymph node. Now, as this fluid percolates into the subcapsular sinus, this then is going to flow down beside the trabeculae. You can think of these as cortical sinuses because they are flowing through the cortex, but more appropriately, they're going to be paratrabecular sinuses. And then from the paratrabecular sinuses, as this fluid's percolating in and throughout these areas around our leukocytes and lymphocytes, this fluid's going to make it into the medullary sinus, where it will then leave the lymph node at the hilum and enter into the efferent lymphatic vessel. So once again, we've got our capsule here. We've got our subcapsular sinus. And you can see sometimes it may be more obvious than others. You've got a lot of lymphocytes here. And then here's one of the connective tissue trabeculae diving down in. And this is going to be our paratrabecular sinus as this fluid is percolating through here. But the fluid is also percolating all through these greater densities of cells as well. This is not solid tissue. So these cells are being bathed in the lymph coming into the lymph node. Here we have a low magnification view of a lymph node cut in cross section, and you can see the green arrows representing the flow of the lymph in our subcapsular sinus. Can't really see our paratrabecular sinuses here in the cortex, but that fluid is flowing down through here. It is flowing down into our medullary sinuses, these large open areas in the middle, and it will be leaving through the hilum. Again, we can't see that. We can't see the efferent artery here in this particular section, but this is the direction of flow, outer toward the middle, and then leaving at the hilum. Now, I mentioned already that we have these reticular connective tissue cells that are helping maintain the shape of our sinuses by and large, but we also have other cells, the stellate reticular cells. Now, these reticular cells are going to be present in the sinuses, but they also are going to be antigen presenting cells. So the macrophages are not alone in their ability to present antigen to our immune cells. Here we have just an example of our subcapsular sinus. Here we have the capsule, have the space. Here we have these larger macrophages, again, sort of a bubbly appearance to their cytoplasm. And here you can see some of these connective tissue reticular cells that are holding their shape here in the sinus. That's their job. The stellate or star-shaped reticular cells, these are going to be the ones that are also the antigen presenting cells, and we are not likely going to see them in this whole mass of cells that we're seeing. Predominantly we're seeing lymphocytes. So we've seen so far, we've got the cortex on the outside part, the medulla in the very center, and this is not unlike what we will see with other organs that we will encounter. And then we have a zone that's in between, sort of a gray area. Is it really cortex? Is it really medulla? And that's referred to as the paracortex. So in this case, we can clearly see the cortex on the outside. Here's our capsule, our subcapsular sinus. We've got some trabeculae diving down, paratrabecular sinuses following those down. And I want to point out these areas. It's almost like you have this dark ring surrounding a lighter center. We've mentioned these before, but these are going to be our lymph nodules present inside the lymph node. And those lymph nodules are going to be present in our cortical zone. Now, the medulla, we're going to have these medullary sinuses. That's how we're going to detect those. And this may actually be a section through an efferent lymphatic vessel leaving the lymph node. And then we have this region that you really don't see lymph nodules. You don't really see much in the way of medullary sinuses. So this is that intermediate zone or no man's zone. We just refer to that as paracortex. So now let's look at the different regions of our lymph node, starting with the outermost portion first, and that being the cortex. Now, as we've mentioned, we have a connective tissue capsule that is going to dive down in toward the medulla of the lymph node, and that's going to be the trabeculae or the septum that will divide the cortex portion up into individualized compartments. Now, within these individualized compartments, we're going to have accumulations of lymphocytes. And we're going to refer to these accumulations as lymph nodules, not nodes. The node is the whole organ. These nodules are these collections, and we're going to visualize them in a couple of different ways. Now, the first type of lymph nodule is going to be a primary lymph nodule. And this is going to be a solid, darker appearing lymph nodule. Now, we're going to have a lot of B cells here. These are naive B cells, memory cells, and these are either entering or leaving this nodular portion of the lymph node. Now, the next type of nodule is going to be one that's a little bit further advanced. This is going to follow an antigenic challenge. So they're no longer naive, but they've been exposed to a particular pathogen. They can now be mounting that immune response, and this is going to look more like a donut. We're going to have a darker outer portion that's called the corona, and the lighter central portion of this secondary lymph nodule is called the germinal center, and that's how you will distinguish a primary lymph nodule from a secondary lymph nodule. So as we look in our illustration, we can see the cortical tissue. Here we have the medullary tissue. The septa will divide and subdivide the cortex into the compartments, and you can see they're not complete. They don't go all the way down, so that the medulla is continuous, one area with another. And it's going to be in this region that we find a lymph nodule. Now, this dark ring is, someone's drawn that in. It's not actually going to look like that. But if we actually look at a slide, here we can see our capsule. We've got our subcapsular space here. Now you can see this more dense ring. This is going to be our corona, and the G represents our germinal center of a secondary lymph nodule. Now, within this germinal center, we're going to have three different zones. We're going to have the dark zone, the basal light zone, and then the apical light zone. So as we go through these, what we're going to see is that the dark zone is actually the one that is most basal. And so the basal light zone is kind of a little misleading because it's not the bottommost of our zones. But the dark zone is going to contain B cells that don't have their immunoglobulins expressed yet. We call these centroblasts. So you can think of them almost as naive. They haven't been exposed, but they can't recognize immunogen because they don't have them expressed as receptors on their cell surface. Now, in the basal light zone, as we move upwards, we're going to see that these centroblasts migrate into this zone when they start to produce those receptors on their cell surface. And it's this light zone, and it's light because the cells are larger. These are the follicular dendritic cells. We also have macrophages there. And so these larger cells, less dense region, give us this lighter appearance. Now, the apical light zone is where, again, these centrocytes is where they're moving up, and they mature, and they're either going to become memory cells or plasma cells, so they're leaving the B cell past behind. And now they're cloning, mounting either a primary or secondary immune response, and as they become memory and plasma cells, this is where they leave the nodule and enter into the circulation. So here we have a secondary lymph nodule. Here we have the dark zone. They call this the light zone. This is the mantle zone they're talking about around, or the corona. And this light zone we divide into the basal light zone and the apical light zone. That's just not illustrated here with labels. Once again, we can see some of the cells that we have present inside the germinal center. The smaller, darker cells are going to be our lymphocytes. That's mostly nuclear material we're looking at. Then we have the larger, much more cytoplasm, macrophage. Here we have our follicular dendritic cells. These are also the antigen-presenting cells, much like the macrophages are. And then some of these reticular cells that are here for support only, they look fairly squamous in nature. Those are going to be the reticular connective tissue type cells present and supporting the structure of the lymph node and nodule. All right, so in the cortex is where we find the lymph nodules. Now, as we move deeper, we get into this region, no man's land. It's not cortex. We don't have lymph nodules. It's not medulla. We don't have medullary cords. But it's in this paracortex that we primarily see the T cells. B cells were up in the nodules. Now we're seeing the T cells. We also have antigen-presenting cells in this region activating and combining with our helper T cells. And as these T cells are migrating into this area from the medullary, to the medullary sinus, they are going to find their way of exiting the node after they have been antigenically challenged by the antigen-presenting cells. They're going to move their way to the medulla so they can leave, get out into the circulation, and get to the area of an infection. Now, it's within this zone that we're going to find those high endothelial venules. Again, these small diameter luminal structures that are going to have the cuboid-shaped endothelium present inside in the tunica intima, and this is going to be the location where we have a lot of diapedesis of our cells in and out of the circulation. So here we have two examples of our high endothelial venules. They don't look as cuboid shape as you might see in some, but they are certainly not the real flattened squamous tissue that we're used to seeing when we look at capillaries and venules. And here we have the other examples, and there in the upper illustration you can see we do have a leukocyte in the lumen of this high endothelial venule. Now we get to the deepest portion of the lymph node, and this is the medulla. And here we have lymphatic sinuses, and these are going to be spaces between our cellular clusters that are medullary cords. Now, all types of cells are going to be here. B and T lymphocytes, plasma cells, memory cells, macrophages. They're all organized into these cords by the reticular fibers and reticular cells, and between those, simply the spaces that we have are our medullary sinuses. So in our illustration in the medulla, the white areas are the sinuses, and the darkly staining areas, much more dense because we have all of our cells accumulated here. These are our medullary cords. So once again, a little bit higher magnification, we can distinguish this loose tissue of the medullary sinuses. We see the accumulation of cells in our medullary cords. You can see that we have our blood vessels present in the medullary cords as well. And the pink fibers that are out in the medullary sinuses, and also within the cords we can't see as well, those are going to be the reticular cells made up of collagen, reticular cells also depositing this material. Just another illustration of our cords showing an accumulation of macrophages. So we do in fact have antigen presenting cells even down in the depth of the medulla. Now with our vascular supply, we've got to have blood vessels. So it's not just enough to have the lymph coming in, because that's really, oxygen deprived. So arteries enter the lymph node at the hilum. They're going to course throughout our tissue from the medullary cords as we get up to the cortex, where we're going to have our capillary beds in the cortex. Then our postcapillary venules we're going to see present in that cortex as well as we begin to send blood back down through in our venous system. We're going to have our postcapillary, we're going to have our high endothelial venules down in the paracortex before we start combining them with larger and larger veins until we get to our vein that leaves also at the hilum. So here's our flow of circulation. The artery coming in for our lymph node, smaller and smaller diameter till we get into our capillary bed in the cortex. Our postcapillary venules will be in the cortex, high endothelial venules down in the paracortex, and they will join together in larger and larger veins that also will eventually leave the lymph node.